Hi everyone, it is lovely to meet you all virtually. As Kirsty said, my name is Louisa. I am a cancer researcher from the University of Nottingham, and thank you for inviting me to speak to you today. So I usually would do talks like this in person, so I want to apologise in advance for any technical issues. Despite being a scientist, technology and computers is really not my strong suit, so hopefully we can get through this without any technical mishaps. So today I thought I'd talk to you about what it's like to be a scientist and about some of my day-to-day work as a cancer researcher. And after that, we'll have some time at the end for questions. So if you think of anything at all during the talk, please just let me know and we'll have a chat about it at the end. So my research is based on brain cancer, and I specifically research some of the mechanisms that cancer cells might use to make anti-cancer drugs or chemotherapy slightly less effective than it should be. But before I talk to you about my research, I first wanted to start by asking you all to think about what a scientist is to you. So if I asked you to imagine a scientist, or maybe draw one without thinking about it too much, what would you draw? So if you'll just have a think. And now by putting your hands up, would anyone draw someone quite old, maybe? So if you just put your hands up, I can't actually see you at the moment, but if you look around, then yeah, hopefully you can all see each other. Would anybody draw maybe someone with kind of crazy Albert Einstein hair? Or perhaps anyone with a kind of a white lab coat holding an exploding test tube? So if you did put your hands up for any of those things, I'm not surprised if you'd imagine a scientist looking like that, because this is kind of what we get shown that scientists look like. These cartoons are kind of what I always imagined a scientist looking like at school. So we have the classic Albert Einsteins and Charles Darwins, their big hair, big beards. But in actual fact, scientists do not look like these people at all. So this is my lab group full of real life scientists. So we're all different ages and genders. We're from different countries and from different cultures. And that's actually really, really important in science, like in all professions. Having people from different backgrounds is so vital. After all, when it comes down to it, and what I'm going to try and explain to you today, is science is really just problem solving. So it doesn't matter if you're a doctor, or if you're an engineer, or if you're a cancer biologist like me, your job is just to break down a difficult question, consider different options and angles, and find an answer. If everyone had the same experiences, do you think we'd be able to solve any problems? Of course we wouldn't. So science is really just solving problems. That's all it is. And I'm aware that that might sound like hard work, or maybe even a bit boring, but solving problems to me is exactly what makes science so exciting. The problems that we solve are cutting edge. They are the issues that are facing our society, both today and tomorrow, and they're frequently issues that affect hundreds, thousands, sometimes millions of people. So I put some of the problems I think that we're facing as a society today on this slide. So just some of them that came to the top of my head, things like cybersecurity. Now we're doing so much online, particularly during lockdown. We're all on our phones, on our computers a lot of the time. Things like treating diseases like I do, the climate crisis and global warming, and things like clean energy, making sure that we can travel in a way that's not going to harm the environment. And perhaps one thing that you think of is food shortages. So I'm sure that you're all aware that with a growing population, a huge challenge that we face is food shortages. How do we make sure that the 7.8 billion inhabitants of the Earth have food security? They can eat enough to sustain them and get all the nutrients and all the vitamins that they need. So scientists are doing loads of research in this area. Plant scientists are genetically engineering crops such as corn to make them drought resistant, maybe make them grow more or have increased nutritional value. Some of you might have also heard of scientists around the world growing meat in a lab. So the thought behind this is that once optimized, it could avoid the environmental impacts of meat farming. So put your hands up, would anyone here actually eat meat that was grown in a science lab? and who finds the idea a bit too disgusting? So I probably would eat lab-grown meat, I think. I'm vegetarian, so the idea that I could eat meat without having any kind of environmental impact is quite exciting. And perhaps an obvious kind of example that we're facing as a problem for scientists today is global pandemics. So pandemics have always affected humans. You might have heard in your history lessons about bubonic plague, also known as the Black Death. So this was an infectious bacterial disease that spread rapidly hundreds and hundreds of years ago in the 14th century and was transmitted to humans from small mammals by the bite of an infected flea. But with humans today travelling more and living far closer together than we ever used to, pandemics can spread really quickly through our society, as we've seen with COVID-19. And scientists are so important, both to advise countries on how to respond to pandemics, to develop treatments to manage the disease, and to run testing programmes. So the building that I work at the University of Nottingham has a testing programme to test university staff and students and monitor for local outbreaks of the virus. So we mostly test for COVID by looking for high quantities of a sequence found in the COVID-19 virus. And we run these tests in plates. Hopefully you can see my camera, but we run the tests in plates that look like this. So hopefully you can see this. This is a 384 well plate. So this tiny plate that's about the size of my hand has 384 tiny wells. And this is what we usually run COVID tests in. This is obviously an unused one. So as you can imagine, you need to have quite good eyesight and a very steady hand to set up reactions that small. You might have also seen in the news that there are a couple of vaccines that now have performed really well in the last phase of clinical trials, which is when you have thousands of people to test that the vaccine is both safe and effective. And these could be available in the UK soon. But what's amazing about this is the scientists that have been involved in their development had to respond really quickly to a new virus that we only actually found out the genetic sequence for, so the information that makes the virus what it is, in January this year, and now they've created something that will likely save millions of lives. So hopefully by this point, I've managed to convince you how exciting science can be. I love being in a profession that is constantly evolving and moving forward as we find out more. Although, if I'm going to be entirely honest with you, I never actually wanted to be a scientist. In fact, thinking back to being at school, I don't think I ever really was told that being a scientific researcher was a job. I think I was always told that if you like science, you could be a doctor or a dentist or a vet. And at the time, that was fine because I wanted to be a vet. I loved animals, and from a really young age, I just had my heart set on being a vet. Now, obviously, being a vet is a fantastic career, but the problem with it is, like trying to be a doctor, it's quite competitive and can be quite tough to get into. And unfortunately, I didn't get into vet school. I also found my A-levels quite stressful and tough, and I didn't get good enough grades to even reapply the next year. So the reason that I wanted to tell you this is to show you that sometimes things don't always go quite to plan, and sometimes you just need to take a chance and try a slightly different path. So I ended up going to the University of Nottingham to study biochemistry, which is a mixture of biology and chemistry. And if I'm going to be honest, I wasn't very excited about going. I felt like it was just kind of a backup option for not getting into vet school, and not a very good one at that. But I gave it a go, and to my surprise, I really loved it. I loved the lectures and I found them so interesting. I actually paid attention to them, which I kind of always struggled with a bit at school, was kind of paying attention to the whole lesson. I also really love being in a lab and doing experiments. I think we often think of science as being very kind of book and learning based, but it's actually really practical. Like today and this week, I'll spend about 6 hours a day just in the lab running around doing experiments, not just sitting at a computer, which is something that I really love. I especially enjoyed learning about the biology behind different diseases. Learning about cancer in particular really interested me. My dad had cancer when I was really young, and the care and the treatment that he received, which wouldn't have been possible without scientists and their research, really motivated me to pursue a career in cancer research. So I stayed on for an extra year to do a research project looking at cancer cell invasion. So this is how cancer cells kind of move around the body. And this might be surprising, but the project actually used flies to study cancer, fruit flies. So, you know, the really tiny, tiny flies that you might see around your fruit bowl. it used those. And this may seem strange, but flies are actually really similar genetically to humans. So about 61% of the genes, so the things that are involved in diseases in humans, have actually a recognizable match in flies. So yeah, I did a whole project looking at flies. So between experiments, we kept the flies in these kind of tubes. And they may look a bit disgusting, but the stuff at the bottom is actually their food, which we made in the lab out of maple syrup and apple juice and things like that. And the flies lay their eggs, and then as the eggs hatch into larvae and turn into flies, they kind of crawl up the tubes. So yeah, I did this project and I just loved doing the research. I found it really interesting. So after that, I decided to stay on to do a PhD, which is what I'm doing now. And a PhD is a 4-year research project, and at the end you become a doctor. So again, I hit a few bumps along the way. I actually applied to not 1, not 2, but 12 different PhD projects and got rejected from all of them before I finally got accepted. So there were a lot of times, obviously, that I thought, why was I even bothering? But I didn't give up, and I kept pushing forward and kept sending out applications, and eventually I got accepted into one, which brings us to now. So as I said, my current research area is brain cancer. So I wanted to explain a bit about kind of what cancer is before I spoke about my research problem. So all cancers, and this is regardless of where they are in the body, arise from when abnormal cells grow and spread really, really fast. So we're all made up of cells, 30 trillion cells, in fact, which is a lot of cells. And with that many cells in our body must really tightly regulate their actions when they divide, when they die, and when they move. And this is obvious because we all are a particular size. If our body didn't keep on regulating this, we grow and we would get taller and taller and taller. So our body must tightly regulate the actions of our cells. So normal cells divide, but stop when they are told to, which hopefully you'll be able to see in this video here. So if it loads, you can hopefully see, and this is a real cell dividing. So we have one cell that's dividing into 2. So I'll just play that for you again, if I can. Flick back. So obviously this takes a very long time. So this whole process actually takes, you can see, about 150 minutes. And then the cell has been told to stop dividing, it stops dividing there. Cancer cells, on the other hand, can kind of reprogram how much energy they need to use and can continue to divide and grow out of control without stopping and without dying when the body signals them to, which you can see in this animation. Now, as they continue to grow, they form clumps of cells called tumours. And cancer cells are quite aggressive, and a growing tumour can actually destroy some of the normal cells around it, which can make someone feel really poorly, especially when the tumour is growing somewhere like the brain. When we treat cancer, and even brain cancer today, and we do treat cancer very well today, treatment has just come on leaps and bounds thanks to the research done by scientists and doctors today. So when we treat cancer, we usually first remove the tumour by surgery. We then give patients anti-cancer medicines called chemotherapy to kill any tumour cells that remain in the body. And chemotherapy is basically just a drug that targets any rapidly dividing cells. So any cells that are quickly multiplying, this drug will target. And as I've just shown you, cancer cells divide really quickly. And this means that although chemotherapy is very effective, it can also sometimes make somebody feel unwell, as it is not just specific to cancer cells, but any cells that are dividing quite quickly in the body during that time. So my research problem, and what my research is focused on, is looking at why the chemotherapy drugs we use in brain cancer are sometimes not as effective as they should be, and how we can target different things in cancer cells to avoid using so much of the chemotherapy drugs, with the hope that we can avoid some side effects that we currently see by using chemotherapy. So in a nutshell, that's my project and that's what I study. So to do this, I grow cancer cells in a lab, and then I use them for lots of different experiments. So I thought I'd talk to you about growing human cells in a lab now. So, as I said, I grow brain cells in the lab. So these cells are human and they've been isolated from tumours when the tumour has been removed by surgery. So firstly, does anyone know what the internal temperature of the human body is, if you just have a think? So if you don't know, it's actually 37 degrees. So actually really, really warm. If you imagine a 37-degree day outside, that's a very, very hot day. And this is what cells like to grow at. They like to be at body temperature. So we can't just grow our cells at room temperature. We have to grow our cells in very large incubators, which are set to 37 degrees. So this is a picture of one here, and this is the inside of one here. You can kind of see me in the background. Now, most cells are happiest when they're growing on a surface of some kind. So they like to be able to attach and spread. And you can tell this just by looking at us. We're solid, aren't we? We're not kind of floating around, our cells aren't floating in the air. Cells like to attach onto one another and onto surfaces. So we grow cells in flasks, again, I hope you can see my camera, like this. So depending on the cell line, a flask this size can contain between 1 and 6 million cells. And these flasks are treated with something that the cells really like to attach to. So I think it's amazing that something, again, size of my hand, can have 6 million cells in. So these are a picture of some of my cells here. So this is taken at 40 times zoom. So it's 40 times more magnified than you can see just with the human eye. Now, cells can look a bit boring just under a normal microscope, but what we can do, which is quite cool, is we can stain the cells with different fluorescent dyes to detect different things that we might be interested in within the cells, which you can see in these pictures here. So if I just try and zoom into this, so you can see it a bit better. So here are some of my cells, which I've stained with different dyes to look at things that I'm interested in. And to take these pictures, we use very large, very expensive microscopes called confocals. And these microscopes use laser beams to be able to detect all the fluorescent colours that you can see in this picture. So this, I think, makes the cells look really quite beautiful. You can see them there as well. There we go. So obviously we need to grow cells in something. They don't like just being on their own. They're used to being inside a body. So what we grow cells in is something called cell culture medium, which, funny enough, is a bright pink liquid. And it is a solution that contains all the nutrients that cells need to grow and to survive. And when we do any experiments and we look after our cells, because we need to look after them kind of every day, every couple of days, we do it in something called a cell culture hood. So you've probably seen pictures of these in the news at the moment. So these can have a gap underneath a glass screen where your arms fit. If you are using more dangerous cells or working with certain diseases, you might have hoods where you kind of put your arms into kind of glove type things to add an extra layer of protection. But basically, we culture things in a hood and not just outside in a lab, because we need to protect the cells from us. We don't want to infect them with bacteria, we don't want to breathe on them, and also to protect us from anything in the cells. So we culture them in a hood so both the cells and ourselves are completely protected. And then one really cool thing that I think is one of the coolest things about working with cells is how we store them. So obviously, if you want to go away on holiday or you want to store your cells for a really long period of time, we need to store them in some way. And what we do to do this is use something called liquid nitrogen. So I don't know if you've heard of liquid nitrogen before, but it is a liquid with a boiling point of minus 196 degrees C. So it is very cold. If you imagine that water boils at plus 100 degrees, this is obviously a lot colder than that. And we use this because I suppose the best way of explaining it is, I don't know if you've ever seen the old Star Wars films where they freeze Han Solo and then they defrost him again. And he hasn't aged, he looks exactly the same. This is what we need to do to our cells. We need to be able to freeze them, and then when we thaw them out again, we want them to be exactly the same as they were when we froze them. And this is why we use liquid nitrogen. It's so cold, it allows us to thaw them, to stop them dividing, and to stop them, I guess, aging. And then when we thaw them out, they're exactly the same, and we can continue to use them. And this means that we can keep cells for years and years and years. So another thing that I wanted to talk to you about today is the way that some scientists can use viruses in the lab as part of their experiments. So we've already spoken a little bit about the COVID-19 virus today, and for many people, obviously and understandably, viruses are a really, really scary word. But I thought you might find it interesting to learn that we also use parts of viruses that obviously have been adapted so that they're safe to use in the lab to do experiments. So one thing we know that viruses are really, really good at is infecting cells. And what they do is they insert their viral DNA into the cell that they infect. So I don't know if you've heard of DNA before. If you have, you'll know that DNA stands for deoxyribonucleic acid, quite a long word, so that's why we use DNA instead. And DNA is just the material that carries all the information about how a living thing looks and functions. It does this by telling our cells to make different things called proteins, and these proteins in turn help the cell to do its role in the body. So DNA controls everything from what colour your eyes are, to whether you can roll your tongue, to what colour your hair is, how tall you are. And we as scientists use the fact that viruses are very infectious and can insert viral DNA into the cells that they infect. And we can then use the viruses to make the cells that we work with behave differently. For example, we can make them produce lots of a particular protein, or we can stop the cells from making that protein at all. And this is really useful as one of the best ways to learn about what a protein does in the cell. For example, in a cancer cell, is to make it either produce loads of it or just to stop it producing it at all. And this is something that I do in my lab to study and learn about proteins that I think might be involved in brain cancer. So one thing that I think is quite cool about this is that we use virus particles that not only change how the cells behave and what they make, but also that when they're inside the cell, they make the cell produce something called GFP, or green fluorescent protein. So I don't know if you've heard of GFP before, but GFP is a protein that was actually originally produced naturally by cold water jellyfish that live in the Pacific Ocean. And scientists saw these jellyfish glowing bright green when they swim, and they realized that these jellyfish must make something in their cells that make them glow bright green when they swim. So being able to mark cells like this not only looks really amazing in the jellyfish, but is also really useful in science experiments. So now scientists use this protein all the time. And for me, by using virus particles that produce GFP, I can tell which cells are successfully infected with a virus as they'll glow bright green, which is what you can see here. So these are my cells before, and these are my cells after. I can see that they're glowing, which means they have the virus in them, which means that the virus is stopping the cells producing the protein, which means I can study what the cells do and if they behave any differently and work out what function that protein has in the cells, which is actually really cool. And I think it's amazing that scientists have adapted a virus, which, as I said, is a scary thing, to use it in a useful way and help kind of our research progress. And I think that's really amazing. So today we've spoken about what scientists do, and I've shown you some of the brain cells that I grow in the lab, and I've told you a bit about cancer research and some of the other experiments that I do. And I hope you found what I said interesting. And if there's one thing that I'd really like you to take away from today, it's that in 2020, scientists do not all look like Albert Einstein and Charles Darwin. Science is exciting and it's modern, and above all, everyone can be a scientist. So thank you very much for listening, and I'd love to answer any questions that you have. I'll stop sharing my screen now, and hopefully I'll be able to see any questions that come through on the chat.
That was lovely, Louisa. That was such a fantastic presentation there. So if anyone has any questions, you can either ask your teacher, we can put it in the chat, or you can say it out loud if you like. So we got one here that says, do you research the effects of drugs?
Yes, I do. So one thing that I am really interested in is looking at what happens when cells become resistant to drugs. So something that I find really interesting in my research is I get cells and I treat them with higher and higher concentrations of drugs, and then look at them and how they act now they're resistant. Because one problem that we have in lots of diseases is that cells can become resistant to the drugs we use to treat them with. which means that sometimes we can't treat the diseases as well as we'd like to. So yeah, I try and make resistant cells, and then I look at other ways that we can kill them and other ways that we can treat the diseases. So yes, I do. And then other people can look at kind of new drugs and do new drug screens using cancer cells, but that's not my research area, but other people in my lab do that as well.
Would the cells look differently if scanned under UV light?
So I'm sure they would, yes. So actually UV light is something that would kill cells if it's a high enough kind of concentration of the light. So you can do experiments to see to kind of mutate DNA using UV light. So the DNA in particular, if we really zoomed in and thought about it from that, would look very different under UV light. But cells themselves, I've never looked at them under UV light, but I'm sure they would too. But yes, UV light is something that would change your DNA and mutate your DNA.
Mm-hmm. Can eating certain fruits like tangerine and oranges heal cancer?
I think with all cancers, and if you're thinking about just being healthy, obviously you want a really balanced, healthy diet. You want to be eating lots of fruit, lots of vegetables. But I don't think eating certain fruits can necessarily heal cancer, but it's always really important to eat lots of fruit, lots of vegetables, to be healthy and keep your body healthy.
Is cancer when your body fights your body?
Oh, so that's a really good question. I suppose in a way, I've never thought of it like that. But yeah, that's a really, really good question. So cancer is when a cell's DNA changes slightly and it mutates. And then that cell, which obviously was a normal body cell, starts acting differently. It starts acting abnormally and it grows quicker, divides quicker, and it doesn't die, which is like the animation I showed you. And yes, that cell did originally come from a normal cell in your body. So in a way, I guess you could, look at it like your body is fighting your body, yes.
How long can cancer last, and what is in the medicine that cures cancer?
So cancer can last, it depends on what kind of cancer you have, and the treatment and lots of different things. So that can be lots of different times. And I'm not a medical doctor, I'm a research doctor. So medical doctors are probably the best people to ask that kind of questions too. What's a medicine that cures cancer? So yeah, we use lots of different medicines. So as I said, the first step is usually removing a tumour by surgery. And then we use things like chemotherapy to target any quickly dividing cells. There's also kind of new medicines that are always coming through because of kind of the research that scientists and doctors are doing. So things like that. One kind of medicine that's exciting at the moment is kind of reprogramming the body's immune system to fight cancer cells. Scientists are also working at using viruses that they've kind of modified as a way to target specific cancer cells and leave the body's normal cells unharmed. So there's lots of different medicines that are always being kind of made and adapted from other diseases that we're kind of using to fight cancer. So there's lots and lots of things.
I've got someone here asking for some tips on how to work hard to become a doctor.
So, as I said, there's kind of two types of doctors. So there's the doctor that I'm trying to be, which is a research doctor. So we're based in labs and we do lab experiments. And then you have medical doctors, which is what most people would know as a doctor. And these are the doctors who you see at the GPs, who are doing surgery, and who you'd see if you actually go to a hospital. So I think for both cases, it's just being tenacious, it's really working hard, it's not taking no for an answer, it's keep on studying, and it's being passionate, I think, for what you're working on. It doesn't matter, I think, how good you are at something. If you're not passionate about it and you don't enjoy it, then it's going to be really difficult to kind of reach your goals. So it's making sure you really love what you do, and then I think everything just falls into place after that.
So how many types of cancers are there? I can imagine there's a lot.
So there is a lot of different types of cancers, but I think I would probably say that every person has a different type of cancer because we are all slightly different genetically. So even though there might be hundreds of set types of cancers, like brain cancer, breast cancer, etc., I would say that every single person has a slightly different kind of cancer. So it's as many different people who have cancer, I would say, is as many cancers that there are.
Can you get lung cancer even if you aren't exposed to smoke?
So yes, you can, but smoking greatly increases your risk of getting lung cancer. But yes, you can still get lung cancer even if you haven't been exposed to smoke.
And can you get viruses twice? And does a healthy diet help with viruses?
So I think, especially this is something that I think we're talking about a lot in the news at the moment with COVID, is if you've had COVID once, can you get it again? And I think it depends on how long your body and your immune system can remember how to fight that virus. That's the first thing. So it depends how close the exposures have been to one another. And also viruses, like bacteria, are mutating kind of all the time, and if it's mutating, it's slightly different, or it's a different, what we call a strain of the virus, then you probably would be able to get that one even if you've had a different strain of that particular virus before. And does a healthy diet help? A healthy diet helps with everything. So yeah, always good to eat your fruit and vegetables.
What's the most dangerous cancer?
I think all cancers are probably quite dangerous. I think it depends on the person who's getting them, the age of the person. So there's a lot of different factors. So I wouldn't want to say that one cancer is particularly more dangerous than the other. I think there's a lot of different factors involved.
What reactions can you have to brain cancer?
Right, so because of where brain tumours are, obviously they're in your brain, and your brain controls your movement and other things like that. People can often feel very dizzy and very sick all the time. So they're kind of the symptoms that you often get if you have brain cancer, is feeling dizzy, feeling really, really sick, just feeling really unwell, because obviously your brain is such an important part of your body.
Yeah, I think that's all. So if anyone else has any last minute questions, now's your chance. But that was wonderful. Thank you all P7s for taking part today and asking lots of really good, interesting questions. And a massive thank you to you, Louisa, for joining us today.
Oh, thank you for having me, and thank you for all the questions. All very good questions.